got into study of drug addiction because I'm interested in the question of how does the brain make choices, what does it mean for the individual to be different, why are we different, what are the genetics, how does the brain work, what's involved with our conceptions. And I got interested in trying to understand that from a very reductionist approach. My attitude first as a graduate student was kind of frustration with some global psychology theories that really didn't take into account the actual workings of the machine and how the brain, neurons and transmitters and all of those pieces came together to work on a functioning bit. I mean, it's fine to ignore the details if you're using a laptop computer. You don't need to know how to build one in order to be able to use them in a creative and effective way. But for those of us who have our laptops fail, we want to be able to know how to fix them. And you can't fix them by just banging them on the side of the case, hoping that something will get back into place. You need to understand how the individual parts are working so that you can pop in the correct board when it doesn't work to be able to reboot it. I mean,
you make me laugh because I'm thinking that most people throw their computers away when they don't work and they get a new one. Maybe that's what society does.
Well, if we could do that with people, that would be different. We can't do that with people. We can't do it. That's the point. Is we can't just toss them out and buy an upgraded version. And so we got to figure out how to fix them. And so we can't fix them if we don't know how the parts come together and how they work. And that means you got to start with a reductionist approach where you understand how the brain is built. And so I started with a reductionist approach thinking about neuropeptides and neurotransmitters and neural circuits. And one of the things, just kind of to give you the bottom line, one of the things that we found was that the endogenous dynorphin systems are neuropeptides that work in the brain as neurotransmitters. They work on a class of opioid receptor very similar to the receptors that morphine works on. And they work in a way that encodes the stress-induced changes that we adapt to stress that result in feelings of anxiety and dysphoria. That sounds like a risk factor for addiction. Let me tell you a little bit more about how I got there. Okay, so part one of the talk is what are dynorphin peptides, what are the kappa opioid systems, and these are the evildoers in Kube's dark side model. Have you listened or have you read George Kube's review? He comes out with some very, very articulate descriptions about what he thinks is important for the dark side. In previous courses, he'd actually been one of the speakers, but now that he's one of the exalted directors of NIAAA, probably be hard for him to find the time to come out here to do this. But just in a nutshell, does anybody know it? Does somebody want to describe what Kube thinks the dark side is? Something you've read about? What do you think?
Can explain allosteric load?
Yeah, can you explain that, what that means? Well, go ahead.
The dark side is when after withdrawal again, the patient goes to using again?
Yes, exactly. So what he says is that when you look at the natural history of a person consuming a euphorogenic drug like alcohol, makes us happy. We enjoyed a glass of wine last night. It made us cheerful and we thought we were charming and witty. But what happens if you take alcohol to a great extent, you become more and more dependent on it. And the fun of the alcohol kind of dissipates. It kind of gets down to not so much fun anymore. In fact, alcoholics say they don't drink because it's fun anymore. They drink mostly to avoid the withdrawal phase. So if they stop drinking, the dysphoria and the anxiety that they get during the withdrawal phase is what motivates them. The negative reinforcement of the withdrawal is what motivates them to consume drug. And that's a switch from recreational now to kind of a self-medication element. Not strictly compulsion at this stage, but the individuals are self-medicating by consuming alcohol, not because it's a wonderfully euphoric drug anymore, but because it's what they need in order to stay well. You hear the same thing when you talk to people who are opiate addicts. You talk about heroin addicts, and you listen to what they're saying about their experience. They start out feeling, wow, heroin is the best drug ever, it's orgasmic, it's really wonderful. They can't wait for the next injection. But as their use goes on and on, what you find is that they're mostly struggling to try to get more heroin to avoid that sickness feeling that they get in between doses. So what is that sickness feeling all about? Koob and I both believe that it has a lot to do with the dysphoria that's encoded by the stress-induced release of endogenous dynorphins. And when the dynorphin systems are turned on, it's part of the adaptive portions of the brain that help signal something's bad and you, the individual, have to get up from where you are and go do something. For a heroin addict, it's go get more heroin. For an alcoholic, it's go get more alcohol. But you might wonder, well, why do we need dynorphin? Wouldn't our lives be so much happier if we didn't have this peptide? Yeah.
I'm not sure if withdrawal syndrome in alcoholics is close to hangover in non-alcoholics, or because hangover is rather metabolic protein, right? Of alcohol metabolism, but the metabolites are.
The answer is that there are certainly elements of the hangover that are manifest in someone who's alcoholic and they're in their withdrawing stage. The severity of the withdrawal phase, the qualities, the physical reaction, the physiological reaction are similar, but it's much more severe. The more tolerant you gain, in other words, if you binge for 3 days where you've been blind stoned for 3 days or a week, the degree of tolerance that you've developed and the crash, that delta is huge. If you just go on Friday night and you drink too much, you feel lousy Saturday morning, but that's a smaller delta, but if you can imagine multiplying that by an order of magnitude, that's what you get when you talk about an alcoholic withdrawal after a binge. So that's my impression.
You're talking about some of the physiological changes, but there's all the sorts of psychological ones, like, you die from stopping alcohol, right? The benzos, you get seizures.
I also feel that the bigger hangover, the more protected as in ancient.
That we're talking about the protection of this form, but that's a.
Yes, I think that's right. I think that there is some, so if you're smart and you realize that, well, I feel sick on Saturday if I drink too much on Friday, maybe next Friday I won't drink so much, then you've made the maturation step that most of us do, and that we don't go in and get drunk to the point of vomiting. Often, we just don't do it because we realize that it's pretty unpleasant. We smell bad, our friends think we're stupid, we crash our cars. There's all sorts of negative reasons why we don't drink to vomiting. But yet, alcoholics have gotten past that. They've gotten past it. They have all of that, but they don't learn in the same way not to do it. They've gone to a different stage. They put up with that. But I was asking more of a ethological question. Why do we have a system like dynorphin? It's exactly that way. It's a clue that we did something bad on Friday night, and if you listen to that clue, you don't do that behavior anymore. It's a way to shape your behavior in a way that's appropriate. It's a reaction to chronic, inescapable stressors, and so most of us learn when we're in a situation where we have stressors that just make us feel awful, we change jobs, we get a different girlfriend, we move out of our house and go away from the kinds of stressors that we move, we do something. But so we need a system like that to enhance our survival. But if it's a manifestation like the dark side, then it's really one of the motivations to drink again for the people who are opiate dependent or alcoholic. And so let me back up and give you a little bit of description of what the dynorphins are. Again, if this is all familiar to you because you've heard it before, I can go through it pretty quickly. But it was really a pretty exciting period of science. I think Chris and I were both involved in the discovery and the development of our insights about how the endogenous opioid systems were structured and how peptides were synthesized and how they worked on opiate receptors. And we now know that the dynorphin, the endogenous opioid peptides, were first detected in the early 1970s as extracts from brain. Hans Kosterlitz, John Hughes found in the enkephalins. The beta endorphins were discovered in again as well in the lab that Chris was in, and in the lab that I was in, Avram Goldstein detected dynorphin in pituitary extracts. And so we now know that there are families of endogenous opioid peptides that work on these receptors. The dynorphins are derived from a single precursor molecule, a signal sequence, and then there's a sequence, the alpha neoendorphin, dynorphin A, dynorphin B. That's kind of cool. So one precursor molecule is cleaved into 3 different active forms of the dynorphin sequence. And the 3 different forms share structural similarities. You can see that this is a single letter amino acid code for the neoendorphin, dynorphin. They all start with the tyrosine residue at the amino terminus. They have the next 4 amino acids the same. And what's unique about the dynorphins in this group are they have basic residues in their C-terminal extension that confer kappa opioid receptor selectivity. And that's different from the enkephalins and the beta endorphins, which are missing. That gives them selectivity. Question? Okay. And you know, having known the peptides, we went through a period of looking at where they were. Here's an example of an immunohistochemical mapping of dynorphin B, but you can map where the dynorphins are with antibodies, and you can look at how the molecules are processed and how they work. At the time, it wasn't terribly clear how those molecules work as neurotransmitters. Go ahead.
Why is that in the guinea pig there are more kappa receptors than in the rat?
Why? I have no idea. I have no idea.
Has even more.
But it's certainly true. It's certainly true. In humans, we have a lot of kappa receptors, a lot more than rats and mice. So guinea pigs and humans express a lot of kappa receptors, and the reason why we've diverged and how it influences our behavior is not known. Not known. It's very clear that it's true, though.
In the behavior studies, can it make a difference?
Of course. Oh, of course. I mean, if you work with an animal, a model that doesn't express the system that you want to study, you're not going to get any interesting effects. But we can see clear dynorphin actions in rodents, even though their abundance of the kappa receptors are not striking. And we know that the kappa drugs work in rodents in similar ways, not identical ways to the way they work in humans and primates. But there are important differences that you're quite right to pay attention to, because it's very clear that rodents don't do everything, react in the same way in every way, the way humans will. So it's predictive, but not always so. But what happened after the peptides were described and how the circuits were mattered, we started to learn a lot about the way these molecules functioned as neurotransmitters. You're all familiar with the way glutamate works, with the way GABA works. How are neuropeptides different? Well, we asked that question, and it took us about 10, 15 years to answer that question. But essentially, with the way neuropeptides function in the brain are more of a neurohumoral mode. GABA and glutamate, fast-acting transmitters that are on and off. They change the excitability plus or minus in the individual neurons. You can identify the circuits in that way. But clearly, the neuropeptides set the tone. And so the tone can either be stressed, for example, when dynorphin works, or not stressed if dynorphin is not working. And so we know that dynorphins are processed, packaged, and they're contained in large dense core vesicles. Dense core vesicles tend not to be docked the way small clear vesicles are docked at the neural junction. And so they are released only when the cells are hyperactivated. They're hyperactivated by situations in which there's a lot of excitatory input, takes more calcium to come in, and you get a different tone. They have very high affinity for their receptors. So unlike GABA, glutamate, acetylcholine, which tend to have affinities in the micromolar range, these peptides have binding affinities in the low nanomolar range, 1,000-fold lower, and that means these molecules can be diluted about 1,000-fold more, and they can diffuse over a wider range and still have concentrations that are active at the receptors. So they tend to have more of an integrative mode. This has been described as neuromodulatory. I'm not quite sure what that means. We could argue about what that means, but the idea is that they really are neurohumoral in that they set the tone. And a stressed brain that has high amounts of dynorphin released really functions differently than an unstressed brain because of the activation of dynorphin. Okay, so let me move on. We also know that when they activate the receptors, these are standard GI-GO coupled receptors, and so when you do that, you turn on ion channels, the increased potassium conductance. You turn off calcium channels because the G proteins will regulate the ability of the calcium channels to be activated. And we also know that through other signal transduction mechanisms, they activate various MAP kinases, including ERK, P38, and c-Jun kinase. These are famous because they phosphorylate transcription factors. Changes in gene expression are important for the long-lasting effects. Changes in synaptic plasticity are evident, but they also phosphorylate a pretty broad range of other substrates. So we can map all of that, and we can characterize all of that, and we can do all of that. And at the molecular level, we can learn a lot about the way in which these transmitters are functioning. And that sets us up to ask in a more intelligent way, what happens when the animal releases dynorphin and it's working in a way that's helping the brain function. And so at the same time, many of us were at this more reductionist molecular approach. There were a series of studies that were being done in the 70s, 80s, and 90s. Albert Hertz focused our attention on the dysphoric property of kappa agonists. If you have a selective kappa agonist and you ingest it, it really feels lousy. I don't know. There's an herb that people talk about, Salvia divinorum. Has anybody heard about that before? Has anybody tried it? Has anybody tried it? I have not been brave enough to try it, but what Brian Roth found was that the active ingredient in this herb is a really wonderful kappa agonist. It's rapidly metabolized. You don't swallow it, but you make a quid and you suck on it under your tongue. And if you go on YouTube, you can see people who are just hallucinating and describing their experience. It's really a strikingly different experience than if you were to do the same thing with an opiate. It's arousing, it's strikingly dysphoric, it's very hallucinatory, and it's really pretty interesting. It's also analgesic. Okay, but we know the kappa agonists are dysphoric. We know that they are released during stress. We know that the systems are increased and revved up when you take a lot of psychostimulants. So repeated injections of cocaine strongly increase the dynorphin message levels. And Tony Schippenberg and Albert Hertz came up with this idea that what psychostimulants are doing by increasing dynorphin tone is creating an opponent process, part of the compensatory adaptation in the brain, so that the euphorogenic properties of cocaine are opposed by the dysphorogenic properties of endogenous dynorphin, and it regulates our in a compensatory way to restore homeostasis. And based on that, they suggested that our endogenous dynorphin system and kappa agonists would be good therapeutics to treat cocaine abuse. And that was an interesting idea. So when we finished with what we were doing at the molecular level, we did a really very naive, simple experiment. And what we said is, okay, let's see what animals do when you give cocaine, and then ask if you activate the dynorphin release, what happens to the response in cocaine. Easiest way to do that is with condition-placed preference, and that's what I've got pictures of. Actually, you can see a little mousey up here, and here's a stopwatch. The animal is trained with cocaine in one compartment, saline in another compartment. The boxes are balanced, so when they're given a choice, initially they'll go to both compartments, but after a period of training, then you test them in the absence of drug. You develop condition-placed preference, and that's shown here. Normally you wouldn't do it this way, but what we did was we measured the preference after each training session. You can see that the animals are learning to like the compartment that they got the cocaine. And we then stressed the animal, or group of animals. We activated the dynorphin system. We thought that if we activated the dynorphin system and then gave them cocaine while the dynorphin was on board, that it should block the cocaine CPP. We expected it would block the cocaine CPP. Instead, what we got was a doubling of the cocaine CPP. That was really opposite of what we predicted. And whenever you see something that's opposite of what you predict, it just draws your attention. As a scientist, you say, whoa, I can learn something here, what's going on? When we did the same experiment, but with animals that had been given the kappa receptor antagonist, because we knew we were releasing dynorphin, give them the kappa receptor antagonist before we stressed them, what we found is they liked cocaine just fine, but they failed to show the stress-induced potentiation. That's really interesting. Kappa receptor activation by dynorphin release results in a potentiation of cocaine reward. In hindsight, we shouldn't have been expecting that there be a reduction, because I think now we're all well aware that stress increases the rewarding valence of drugs of abuse generally. And that had been known, but we didn't connect the dots until we saw this data. And now we thought that, okay, dynorphin is a stress molecule that's really important in the regulation of reward. So I could stop right here and we can talk a little bit more about stress mechanisms. What would you want to know if you saw this data? If this was your data, you were working in the lab in 2003, what would be the first thing you'd want to know when you saw this?
How about those patients who stay under corticosteroid drug?
I didn't understand the question.
About the patients who stay under corticosteroid drug.
Oh yeah, corticosterone.
Because these are also derivative of the cortisol, and we know that the cortisol has a very effective role in the stress
Right.
situation. How is it?
Well, that's really an important question. And what you're asking is, what's the role of our endogenous corticosterone levels in changing this mood? And I can tell you that for a very long time, when people thought about stressed animals and why they would change their consumption of drugs of abuse, they were focusing on CRF and they were focusing on adrenal corticoids and thinking of those as the mediators. What we find is that you can not see an effect of dynorphin on the adrenal corticoid levels. If you bleed these animals after swim stressing them, the corticoid levels are up as normal, and so dynorphin is not suppressing the glucocorticoids. We have CRF activation of dynorphin release, as well as CRF activation of ACTH release, activation of glucocorticoid release, changes in the brain in glucocorticoids. But those two things are doing different things, and this is a dynorphin response, not a glucocorticoid response. So that's a really important question. What is the relationship between this stress response and other stress responses that we've described? And that's really a very important thing to resolve. What's the relationship between the different stress hormones? What else would you, so you know now that stress potentiates the rewarding effects of cocaine. You think that that's really an important clue because we think that there's a strong relationship to the way humans work. We know it's a risk factor for drug addiction. We think that individuals who are stressed are more likely to abuse drug. What's the next experiment you guys would do?
Could we repeat it in contingent experiments, self-administration experiments?
Okay.
Or the next question would be maybe dopamine. What dopamine is changing?
So phrase that a little bit more clearly. What would you want to know about dopamine?
Is the kappa receptor in that stress situation underlying that?
Mm-hmm. Exactly. So you're asking, how is it that, what is dynorphin doing? Which neural circuit, which system is being regulated? The prediction could be that the dynorphin is causing a change in the way the VTA to nucleus accumbens dopamine signals, so now cocaine is having a more robust effect because of an interaction between dynorphin and kappa receptors in that way. And that's an area of research I can tell you quite a bit about because we did those experiments. That's great. What else would people want to know? What are the neural circuits? What's the relationship between the other stress hormones, stress molecules? What else would you want to know? Yeah. Would a kappa agonist do the same thing? Obviously, stress is a really gross stimulus. A lot of things are going to be revved up when you stress an animal. It's not just going to be dynorphin. What are the other things that are being released? And is activation of kappa receptors sufficient? We know it's necessary. The antagonist shows us that it's necessary, but is it sufficient? That's a good question as well that I can give you the answer to. What else?
Where it is.
Where is it working? You know, if you want to get to the level of the neural circuit, you want to understand where are the stress-sensitive circuits that are controlling the rewarding valence. Where is this actually measured? Where is the animal measuring the dynorphin tone so that it influences the decision-making process? I think that's really has been a lot of fun to address. What else? Okay, so I don't know what order to answer those questions, but let's go and start here with a simple concept. The conception that we had is that animals were entering into the condition plays preference box in a dysphoric state. And now cocaine had a euphorogenic property, but the reason why the animals were responding so strongly was because that delta mood was really exaggerated. The analogy is if you take an antidepressant but you're not depressed, it's not going to change the way you feel and as dramatically as if it works and you're depressed. If you take an anxiolytic and you're not anxious, it's not going to make you feel all that much different, a little agitated. But this idea is what we were thinking, and so we wanted to be able to figure out ways that we could measure dysphoria, because that's what we were believing was motivating the increase in the response to the euphorogenic drug. And it's hard to do. Dysphoria is one of those abstract concepts, much like pain. We know what's painful, but you know, whether my pain is the same as your pain, it's different than if you're measuring the ambient light. You know, you can put out a photoluminometer and you can measure the number of photons that are hitting the sensor. The same stimulus. Okay, I hit my thumb with a hammer, I feel pain. I hit your thumb with the exact same force. Your reaction to that same stimulus may be very different than my reaction to that same stimulus. Dysphoria is similar in the sense that it's more of an abstract concept that's difficult to quantify. What we did was we used a pairing between a stimulus that we thought would be an aversive stimulus and an odorant, a neutral odorant, and then asked to what extent would the animal avoid that odorant. And so if we paired an odorant with an aversive stimulus and then allowed the animal to choose between a compartment that contained that odorant or one that contained a neutral odorant, what we found is that animals would avoid an odorant that had been paired with stress, an odorant that was otherwise neutral, and that effect was blocked by the kappa receptor antagonist. And so the question then would be, okay, so you've got this learning paradigm. To what extent CPP is a learning task? To what extent is this a learning task? We all know that when you're stressed before an exam, you study harder, you remember better, and you really learn better. And when we go into experiences where we change, where we're stressed, we tend to remember those more intensely than if we're not. To what extent is what dynorphin doing influenced by a learning event? The answer is it's not learning. If you're just looking at pairing with cocaine, the kappa receptor antagonist doesn't block the ability of the odorant cocaine pairing. And then the next question we were asking is, where did it happen? So that was a really important question to try to tease apart the nervous system and how it works. And there were two tools that we used to try to resolve that question, to map out where dynorphin was working in the brain. The first tool that we used was the drug NorBNI. NorBNI is an amazing drug. I'm going to come back to it in the second half of this presentation. NorBNI, when you inject NorBNI into an animal systemically, the drug lasts for 4 weeks. It takes a full 4 weeks before the response to that drug is reversed. And that's different from most antagonists. One injection, NorBNI, 4 weeks. And I'll tell you why later, but that's a tool. Now we can locally inject NorBNI into a brain region, and it's like a transient inactivation of the kappa receptors for at least 2 weeks, which is great amount of time to do behavioral studies. And you can ask by mapping where NorBNI injection will result in an alteration of the dynorphin behavior. That's where you can learn where the dynorphin works. Newer tools are available, and we're putting those into our studies now, where you can take a pro-dynorphin message Cre, and you can activate Cre recombinase just in the dynorphin expressing cells, and then with channel rhodopsin you can activate different populations of dynorphin to release dynorphin in ways, and look at the dynorphin circuit in ways that will give you more resolution as well. Another way to learn about where dynorphin is working is to take advantage of the phosphoselective antibody that we developed. When a GPCR is activated by agonists, they're G protein receptor kinases that phosphorylate that cytoplasmic phase at characteristic residues. We identified where those residues were, and what we did was we make an antibody, a phosphoantibody, and you can tell where dynorphin was working based on where those receptors became phosphorylated. I'd spent a lot of time trying to measure dynorphin release directly, and it's really hard to do. You can do a dialysis probe, but it's big and insensitive, and the temporal resolution is lousy and doesn't give you a clear sense as to where it might be working. But this gives us cellular resolution as to where are the receptors. And you can see that in this particular image, you can see that when you inject CRF into the lateral ventricle, there's a big increase in the green staining, which is the kappa receptors that are phosphorylated in the basolateral amygdala. Norbionide blocks the CRF effect, and if you've got a dynorphin knockout, you don't get the increase as well either. And at higher resolution, you can see that the cell types that are being activated, the ones that express the kappa receptors, are on the neurons and also on astrocytes in that structure. So you can learn a lot about where, and you can move your Norbionide injections around in the brain, and you can identify the circuits. We talked a moment ago about how does the other stress hormones relate to the dynorphin function, and one of the ways in which we answered that is what stimulates the release, and it turns out that CRF injection and CRF activation results in dynorphin release, and you can use genetic tools and pharmacologic tools to map that. So we know stress-induced CRF works broadly in the brain. One of the things that CRF does is activate dynorphin release. And what's really interesting is that if you inject CRF, you get a conditioned place aversion. So we know CRF is aversive molecule. It's a stress hormone. What was really interesting is that if you pre-treat the animal with Norbionide, CRF is still acting in the brain, but it's not aversive. Here's a dynorphin knockout animal. Now when you give CRF in the brain, you actually show that it's slightly positive. Interesting. We thought that CRF was the big bad wolf here, that it was the stress molecule. It's not. CRF is really important for the survival of the human. And animals that are missing CRF or missing the CRF1 receptors don't do very well. They die. They tend to not adapt. When we are stressed, we physiologically need to adapt our metabolism, our blood flow, our neural systems in ways that are essential for survival. And that homeostatic, metabolic, physiologic adaptations are really essential for survival. And in fact, we also release CRF when we're happy. You know, you go down a roller coaster, your CRF levels go way up. Your glucocorticoids go up whenever you're aroused in ways that are physiologically meaningful. It's only when the stress is chronic and uncontrollable, repeated, and you develop this dysphoric or anxiety reaction to it. And in the absence of dynorphin, CRF has more of a euphorogenic properties. The story that I like to use to illustrate that is, you know, some of us really like to drive down the freeway at 80 miles an hour. It's really a lot of fun to be zooming along. It's just gorgeous in a nice, powerful car. It's really great. It's a very different experience when you're driving down the highway, but now you're sitting in the back seat and you don't know if the driver's had too much to drink. It's not quite the same experience. Your stress levels are quite high. So CRF goes up in both cases, but encodes different things. This is CRF and CRF in a dynorphin knockout animal, and in a dynorphin knockout animal, CRF is not anxiogenic either. So CRF activation of dynorphin results in a reduction in open arm time in the elevated plus maze, and that's all great. Okay. All of that predicts that if you are stressed, you're going to react to psychostimulant drugs in a different way, and that it's going to increase the risk of drug addiction. And CPP is a great model, but clearly it's investigator-injected drug. It's a passive administration in the animal, and so the animal is really reacting to the experience, but doesn't have any voluntary control over it. But what it does is it predicts that if an animal was stressed, it might self-administer a psychostimulant or heroin or some other drug at a different rate. And we all know that. Again, the story is pretty simple. You've had a great week, you know, you've passed all your exams, you know, you got a paper in Nature, you go on Friday with your friends and you enjoy a beer and it's really a wonderful experience, you're all happy and everything is good. Imagine another week where your paper was rejected yet again from brain research, you can't ever get it published, your advisor's really angry with you, your girlfriend or boyfriend broke up with you, your apartment flooded because the toilet backed up, nothing is going right that week. You go on the Friday and drink the same beer, the same stimulus, the valence of that beer means something really different. And so the concept that addictive substances have their pharmacological activity, but the valence of those drugs and the meaning for the individual can depend upon their context is obvious to us. The question then is, okay, if we give rats the opportunity to self-administer cocaine and acquire a self-administration pattern, we can measure how much they like it, not only by how much they consume the cocaine, but their break point. And we can do that in ways that are pretty sophisticated. I don't want to go through this in much detail, but there are a couple of different ways you can measure break point, but what's really pretty interesting is that animals that have been given repeated force swim stress prior to the self-administration apparatus show an increase in the amount that they value the cocaine compared to animals that are just liking cocaine. Animals that like cocaine, they will administer cocaine, self-administer it quite nicely, but when you start to make it more difficult for them to get the drug, so, you know, they'll just stop using it. Piazza talks quite a lot about that, but you can see here that the price that the animal is willing to pay for the cocaine really goes up, and it goes up in a Norby sensitive manner. That's this data here. Okay, let me just go more quickly because I want to give you a 10-minute break before we go to the second part. And, what I'm interested in as a molecular pharmacologist is where does this work and how does this work and how can we tease that apart. And what I showed you in an earlier slide was that when the kappa receptors are activated by dynorphin, you get this dysphoric effect. If you knock out dynorphin and stress the animals, they don't show the dysphoria. If you knock out the kappa receptor, you don't get the dysphoria. If you mutate the kappa receptor so that a critical phosphorylation site required for G protein receptor kinase activation of the receptor, you don't get the dysphoria. We know that arrestin being recruited by the receptor in a phosphorylation GRK dependent manner results in arrestin dependent signaling. We know that that signaling results in ERK, P38 and Jun kinase activation. And what we found is the drugs that block the P38 MAP kinase block the dysphoria. We know that this is important for aversion. I just summarized the results of 4 papers. Going quickly because the details are things we could discuss if you're particularly interested. If we identify the regions by local injection of Norby, we find that there's a critical spot in the dorsal raphe nucleus that's a serotonergic circuit. And so the question is, how is P38 regulating aversion by regulating the serotonergic input? We used a viral expression approach, and when we used a viral expression approach in a kappa knockout animal, put the kappa receptors back into the dorsal raphe nucleus, we could recover the aversive behaviors even though the kappa receptors everywhere else were gone. We used a conditional knockout approach where we used floxed P38 and selectively excised the P38 only in the serotonergic neurons using promoters that were specific for the tryptophan hydroxylase positive neurons. We did a breeding strategy that allowed us to get inactivation of P38 just in the dorsal raphe nucleus serotonergic cells. These are control experiments that show that. And when we do all of that, we get down to this data which says that if we inactivate P38 just in the dorsal raphe nucleus and just in the tryptophan hydroxylase positive cells, we block the effects of stress. We can do that with a PET promoter or SERT promoter, but when we inactivate the P38 in adjacent dorsal raphe nucleus cells that have the GABAergic, Astrocytes, it doesn't do that. Not only is it important for aversion, but if you socially defeat the mice, you can cause a reduction in social interaction. I was interested in social interaction because I think that aversion as a measure of dysphoria is great, but in terms of trying to translate that into human behavior, this experiment is worth taking a minute to look at. Let's start here. If you take a mouse and put it into the cage of a resident mouse, a bigger, stronger, it's a tome cage, the intruder mouse gets beaten up. The mice understand that they're in the wrong place and they will go away, try to get escape. But if you don't let them escape, they will show really characteristic puppy dog-like behavior where if a big aggressive adult dog challenges a puppy dog, they roll over on their back, signaling to this aggressive dog that they're not challenging. We do this as humans as well. Mice do it, rats do it. And what happens is that if you're really cruel and you take that intruder mouse and it shows the social defeat behaviors, time spent here, social defeat behaviors, but now you do it to that animal repeatedly. You do it to it on a second day, third day, fourth day, fifth day. What happens with wild-type mice is that they increase the amount of time they're showing this behavior. They increase the amount of social defeat behavior. If you pre-treat them with Norby or if you use a dynorphin knockout animal, they show normal social defeat behaviors, but they fail to show this progression. They don't develop this pathologic social avoidance behavior. I think that's really interesting. A lot of us think that's really interesting because if you're trying to think about the disease of depression, what you know is you've got something up here where an individual will interpret a fairly neutral experience and will avoid it just because they imagine, well, it's failed every time I've ever done it. I don't want to get out of bed and get involved. I don't need to go to work because it'll be another terrible day. You imagine that these mice are really feeling like it's going to be another terrible day. And so I'm interested in understanding how that works. And so I'm interested in knowing whether this same serotonergic system is involved. And so when we do this behavior where instead of looking at amount of time shown in the social defeat postures, we allow the animal, as shown here with the red line, to explore a interaction chamber where there is a neutral animal in a box. Normal mice are really curious. They'll go over and they'll explore the new animal out of curiosity or for whatever reason the mouse is thinking. But if you take an animal that's been stressed like this, they will go to the other end of the cage and avoid them. If they get pre-injected with Norby they go back to being normal. And if they have the P38 knocked out in their serotonergic system, they are also normal. And so we begin to get a sense as to what's going on with the serotonergic system, important in this pretty interesting social defeat behavior. Thinking about addiction mechanisms, again, not just depression mechanisms, animals will show cocaine place preference. None of these mutations will affect the animal's place preference to the cocaine. If you extinguish their place preference, you can reinstate that place preference with stress. And in a wild-type animal, you get great reinstatement. But if they have their P38 knocked out, that stress-induced reinstatement is blocked. But they like cocaine just fine, and you can reinstate place preference with a priming. This data simply shows that stress-induced analgesia is unaffected, so it's a fairly selective change. Elevated plus maze data suggests that that pathway is important in the dysphoric, but not the anxiogenic effects of stress. And I've got a lot more data that I don't want to take the time to go through, except to show you this data, which is, how do we think that works? Well, I'm really interested in the kind of the molecular machinery that's involved. And we know that it's going on in the serotonergic system. And we know the P38 is being activated. What this data shows is that what happens is that the serotonin transporters increase their functioning. And so in those projections to the ventral striatum, there's an increase, a significant increase in the amount of serotonin transporter function. And more data, but a cartoon. I want to end up with a cartoon before we take a break. I've got all of the control experiments that'll describe it to you, but here's what we think is happening. In these dorsal raphe serotonergic neurons that are projecting from the dorsal raphe to the nucleus accumbens. The kappa receptors in the medium spiny neurons, the dynorphin in the medium spiny neurons release dynorphin locally onto kappa receptors on those nerve terminals. Kappa receptors there will activate p38 MAP kinase in a GRK and arrest-independent manner. And a p38 mechanism causes the serotonin transporters, which are present in endosomal vesicles, to migrate out to the cell surface where they more efficiently pump serotonin back in. All of that makes sense. We measured that neurochemically, and all of that makes sense because when you think about the role of serotonin in mood, a transient hyposerotonergic state in the ventral striatum makes a lot of sense. And that's the model that we think is important, and I think that I'll stop there, take a break, and then we'll talk more about kind of therapeutic implications of all of this. And other models. We don't understand all of the circuits. We, for example, haven't finished answering the question of how kappa receptor activation in the VTA neurons, also regulating dopamine, also contributes to the dysphoric state. I think that those are topics that I could talk about at length maybe in a break. But why don't we take a 5-10 minute break, go get some coffee, and we'll come and we'll say, how do we use this insight to make new therapeutics for the treatment of stress-induced drug risk? Great.